to Newomics. Welcome to Newomics hosted native mass spec webinar. This is a recorded webinar and will be available for replay later, or if you want to let a colleague know about it. My name is Tom Carney. I'm the Director of Sales here at Newomics, and with me from Newomics, we have Sophie Bianchi, who organized this webinar and is our marketing manager, and our CEO, Daojing Wang, is also here. First, I'm going to give a brief overview of the Newomics multi-nozzle emitter technology and discuss the competitive, or the not competitive, the compatible mass spec instruments and applications. Then we'll have our feature presentation with Victoria Cotham, and after the presentation, we'll have 20 minutes of Q&A. Okay. Now, one thing I wanted to point out here is that the Q&A is a little menu, pull-down menu at the top there. You click on that button, and then you can submit your question at any point during the presentation. We'll answer them at the end. Okay. All right. Thank you very much. All right. So I just wanted to cover the Newomics M3 emitter technology, which is the core technology that Newomics has developed. It is a microfabricated monolithic multi-nozzle emitter. So microfabricated means that it's manufactured with semiconductor manufacturing techniques. And that gives us a few benefits, one of which is it's extremely precisely made. It's a very complex manufacturing process, but it's very precise, very reproducible, which means it's very reproducible emitter to emitter and nozzle to nozzle. So a conventional emitter has a single nozzle. You have a single stream in, a single spray out. What the M3 emitter does is it takes a single stream of fluid in, and then it subdivides it into multiple nozzles, multiple emitters for the electrospray ionization. So the big benefit you get from doing that is by making smaller streams, you're able to make finer droplets, which are easier to desolvate. So it enhances the ionization efficiency of the process, which gives you a number of benefits. One is increased sensitivity, and the other is you're reducing the matrix effect. There's other benefits too, because you have multi-nozzles. If they do start to clog, it's still going to work through the remaining nozzles that are still working. So there's a number of benefits to the technology. So you get the benefits are sensitivity, robustness, and throughput. The throughput benefit is because you can use higher flow rates and still get this great ionization efficiency. Now, to use the M3 emitters, you're going to need one of the Newomics ion sources, the MENC sources, we call it. Here we have one in the picture. That's got a few main features. One is the imaging system, and that's the picture shown down there in that circular inset. I'll get to more detail on that in a little bit. There's also this emitter housing, which allows you to pop the emitter in and out without any tools. And we do have a nano viper connection we use here that you do use a tool for that. We have a specially designed torque wrench because the emitters, as I mentioned, are made of silicon, which means it's somewhat fragile and you have to use the precise amount of torque, but we give a tool, so that's done perfectly every time. So what this system is going to deliver is nanoflow sensitivity at microflow flow rates. It allows for smaller sample amounts. You can enable higher throughput because you can do the microflow flow rates. And it will also give you a higher robustness with the system. Now, the vendors that we're compatible with are the Thermo mass specs. This was the first one we developed in conjunction with Thermo. And we have two versions of it because Thermo has two versions of their mass spec interfaces. One is what we call the NG interface, and the other is the legacy interface. These two instruments cover the entire Thermo product line. And we developed it, as I said, jointly with Thermo. We developed applications with Thermo. They've tested it thoroughly, and we've published joint application notes. And we also have a co-marketing agreement with them. So we worked extensively with Thermo to develop this, and it's available, as I mentioned, on all their mass specs. Earlier this year, we introduced it with Bruker as well, and we went through the same process with Bruker. They, developed and tested the source with us, developed application notes. And we're also doing a co-marketing agreement with the Bruker. So that covers all the Bruker instruments. And the latest one that we've introduced, or are actually going to introduce at ASMS, is the Agilent interface. This one's brand new. We'll be launching it soon. And the co-marketing is not really off the ground yet, but we are working with them, generating data, and they're testing the source out for us. But we'll be producing some applications with them very shortly. So to sum that up, we've got all Agilent, we've got all Bruker, and we've got all thermal instruments for all those mass specs. And here's a close-up of the Mency source. You can see the nanoviper connection here. We'll connect to whatever LC you've got, wherever your column is. And we use a sheath gas which comes from the mass spec. So we also will apply that to the electrospray as well. Now we have a video here. Let me start this. This will show you exactly what an emitter looks like in action. This is with methanol, which gives a nice, beautiful spray. You can see the ionization here, and then the magnetic field pulling it in to the mass spec here. So the reason you need this camera or this imaging system is to optimize the spray. So the spray is dependent on a number of variables, like the distance you are from the mass spec inlet, the voltage you're using, and of course the solvent system. So we have a nice, beautiful methanol spray here. When we're doing native, it's not quite as beautiful. It's more of a thin stream, and Victoria is going to show you a picture of that, I believe, in her presentation. But it still gives you great ionization efficiency. All right. Now, just to sum up the applications that we have a number of application notes on our website. We have some on proteomics. We do bottom-up proteomics, targeted proteomics. We also have intact mass application notes. Reverse phase intact mass. We have oligonucleotide application note using ion pairing reagents. We also have native mass spec application notes with ammonium acetate as the solvent system. And we also have lipidomics application notes. So all these are available for download. Won't go into any more detail on any of those right now. But since this is a native webinar, I also wanted to point out we have a special native mass spec page on our website at www.newomics.com/native-MS, and you can get all the details on the products we have for native and our applications. We recently also introduced a kit here, the Newomics Native Mass Spec Kit shown on the right. It consists of 3 parts. It has a mini table to reduce the risk of high current so you don't get shocked. We have a column set to study the proteins and the protein complexes. So there's 2 columns in the kit. And there's also direct infusion set. So we have a couple of syringes and special filters that you can use for the direct infusion for native mass spec, because it can be a challenging application, and this can definitely help people get started with it much easier. All right, so that's my quick overview of Newomics technology. And now I want to introduce our speaker for today, Victoria Cotham. Victoria is currently a staff scientist in the analytical chemistry group at Regeneron Pharmaceuticals, where she focuses on developing native intact mass methods for the characterization of biotherapeutic protein modalities across various stages of drug development. Victoria earned her Ph.D. in analytical chemistry in 2016 from the University of Texas at Austin under Professor Jennifer Brodbell, where she developed methods that combine ultraviolet photodissociation with top-down and middle-down mass spectral approaches for the characterization of monoclonal antibodies and polyclonal antibody mixtures. Prior to joining Regeneron in 2018, Victoria worked as staff scientist in the proteomics laboratory at NYU Langone Health, where she performed MS-based epitope mapping and large-scale quantitative proteomic workflows. So I'm now going to turn it over to Victoria, and we're very happy to have her here today. Thank you.
Okay, great. Thank you for that introduction. And let me just go ahead and share my screen. Okay. Okay, can I just get confirmation that you can hear me and see my screen?
Yes.
Okay,
Yes.
Perfect. Thank you so much. All right. So Well, first, thank you, Tom, for the kind introduction, and also good morning on the West Coast and good afternoon on the East Coast. So first, I'd just like to thank the Newomics team for this opportunity to share our work on the development of a fully integrated native mass spectrometry approach for the characterization of intact adeno-associated virus particles. And really our overarching goal for this work is to streamline the native analysis process for AAV product characterization within a biopharmaceutical setting. Let me just turn on my laser pointer here. Okay. So over the past several decades, there have been just tremendous advancements towards the development of gene therapies for the treatment and prevention of disease. And in its most basic definition, gene therapy refers to altering the genetic material inside cells. And one of the most common approaches for accomplishing this in vivo is through the use of adeno-associated viral vectors that deliver therapeutic transgenes directly to target cells, which is represented in this figure here. To date, there are more than 150 candidates undergoing clinical development. And really the sizable candidate pool can be attributed to the favorable safety profiles of AAVs, which exhibit lower overall levels of immunogenicity and also vector-related toxicity compared to other commonly used viral vectors, but also due to their stable and long-term transgene expression. Moreover, AAVs are also naturally versatile with multiple serotypes that offer distinct tropisms, which can be exploited for tissue-specific transgene delivery. And they can be further rationally designed through the incorporation of regulatory elements such as specific promoters and enhancers, as well as through capsid engineering techniques that can ultimately increase transduction efficiency and drive transgene expression. So when compared to other very commonly used biotherapeutic modalities, such as monoclonal antibodies, AAVs are significantly larger and more structurally complex. So this fully assembled capsid here possesses a molecular weight of approximately 5 megadaltons and consists of a 60 subunit icosahedral capsid, which is composed of 3 viral proteins, VP1, VP2, and VP3, that assemble in an estimated 5 to 5 to 50 ratio, respectively. And so this capsid creates the protective outer shell that encapsulates the therapeutic transgene of interest. And now what's interesting is that this duality of the AAV assembly really gives rise to a unique set of quality attributes at both the intact protein levels as well as the individual component levels, so meaning the capsid and the transgene of interest. But these all need to be very carefully monitored and controlled throughout product development and production. So for example, at the intact level, aggregation, functional virus titer, and capsid content are identified as potential critical quality attributes that impact the overall safety, potency, and purity. At the capsid level, it's important to confirm the identity of the viral proteins as well as evaluate VP stoichiometry to ensure proper assembly of the capsid, as well as characterize the presence of any post-translational modifications that might be present. And finally, since the transgene is critical for the therapeutic effect, controlling its identity, its purity, and titer are really crucial to the final gene therapy product. So mass spec has emerged as a powerful analytical technique to support the development of AAV-based gene therapy products. And this is largely due to its ability to characterize AAV at different levels. So for example, LCMS-based techniques at the subunit level allow for efficient capsid identification and stoichiometric assessment. And at the peptide level, mass analysis allows us to obtain residue level characterization and quantitation of post-translational modifications. So there's been growing interest in the use of native mass spectrometry approaches for intact AAV characterization, which holds potential as a high throughput and low sample consumption alternative to the industry standard of analytical ultracentrifugation for obtaining capsid content ratios, or the relative amounts of the desired full viral capsids versus those that are either empty or contain partially truncated genomic information. And this represents a critical quality attribute that really impacts the overall safety and efficacy of AAV gene therapy products, and therefore needs to be carefully monitored. But despite the promise of native MS for intact AAV analysis, there are a number of analytical challenges that arise due to both the large size of AAV as well as due to its tremendous intrinsic mass heterogeneity, which arises primarily due to co-occurring viral protein stoichiometries, as well as variations in post-translational modifications and variations in the genomic material. And so to just drive home the point of vast particle heterogeneity, Albert Hecht's team recently showed that AAV capsid assembly follows a stochastic model that's represented by this expression shown here. And what this tells us is that the native mass spectrum for AAV can theoretically represent a composite of nearly 1,900 possible capsid stoichiometries. So as you can imagine, this creates tremendous levels of heterogeneity that have to be taken into account. So now if we consider both the size of AAV and its intrinsic heterogeneity within the context of conventional mass spectral analysis, which generally requires charge state resolution between adjacent charge states in the M/Z space in order to accurately measure mass. So most common mass spectrometers are not optimized for extremely large species and are typically limited to molecular weights around or below 1 megadalton in mass. Above this range, there are a number of variables that start to interfere with mass analysis. More recent state-of-the-art instruments with improved transmission and detection efficiencies, such as the Orbitrap UHMR, have extended this boundary considerably and now allow detection for masses on the order of 10 megadaltons with relative ease. But we still run into limitations for extremely heterogeneous samples, such as AAV in this case, where charge state resolution is not possible. An alternative approach that has gained considerable traction in recent years for intact mass analysis of AAV is charge detection mass spectrometry, or CDMS. And this is a single ion technique that really bypasses the need for charge state resolution to measure mass. And it does so by simultaneously measuring an ion's charge and its mass to charge ratio. And while the adoption of CDMS within the biopharmaceutical industry has been limited by its lack of commercial availability, this technique has provided some key insights about intact AAV ions that can be exploited for native MS analysis. And importantly, what both electrostatic trap-based CDMS and more recently Orbitrap-based CDMS have shown us is that empty, partial, and fully packaged AAV particles possess similar numbers of charges during the native ESI process. And so what this tells us is if the charge remains relatively constant, we can assume that any changes occurring in the M/Z domain arise primarily due to differences in AAV mass. And so based on this assumption, we can start to parse out information about capsid composition versus, full versus empty, which should only really be differing by mass because we assume that their charges are the same. And so we're going to exploit this assumption, empirically derived assumption, for the online method that I'm going to describe today. And so to date, all native intact mass analysis of AAV utilizes some variation of static infusion, like what I'm showing here on the right. So in this case, sample is directly loaded into a borosilicate emitter that's pulled to a very fine tip, which I hope you can see here. And infusion is driven through applied voltage and capillary action. And one of the major advantages of this method is its very high sensitivity, which is achieved due to the small droplet formation that occurs at this very fine tip. And while this is great, this approach does require offline buffer exchange into a mass spec compatible buffer prior to analysis, which can ultimately cause stability issues for AAV when it's removed from its formulation buffer for an extended period of time. But also because of this very fine tip, there often are clogging issues that arise, and this can require frequent tip changes as well as re-optimization of spray conditions, which often vary from one tip to the next. So alternatively, for this method, we're using an online platform that incorporates a small inner diameter desalting column on the front end. And I'm just going to highlight some key advantages of this platform here. One of the first being that we can bypass these tricky static tips and instead use this very robust Newomics multi-nozzle M3 emitter, as shown here. And this helps to overcome any delays arising from tip clogging issues. And also, I just want to point out that in the event that there are emitter changes that are necessary, this can be readily accomplished using the plug and play Newomics Menzi source that Tom just described. This is without any need for additional re-optimization. So it truly is a plug and play type system. Furthermore, this method is very fast with run times of approximately 10 minutes per sample. And this is really significant when compared to the industry standard of analytical ultracentrifugation, which can take upwards of 5 hours just for data acquisition, not including data analysis. And so by optimizing the column conditions, we can achieve efficient desalting to remove non-mass spec compatible buffer components without any need for offline buffer exchange or offline sample handling prior to analysis. And by using really low flow rates here, we can still minimize sample dilution and maximize our sensitivity. One other major advantage of this platform, which is equipped with the Newomics Menzi source, is the ability to perform charge reduction immediately prior to mass analysis using our desolvation gas setup, which I'm showing here, highlighting here. And so just to illustrate this point, let's consider this spectrum shown on the top for AAV8. And this shows the presence of both empty and full capsids. In theory, by decreasing the overall surface charge of the differentially loaded capsids, we should start to see greater separation in the M over Z space based on their relative mass. And so as you can see here in this spectrum on the bottom, we are able to achieve this effect by adding a volatile charge reducing agent directly to our desolvation gas setup. And so now that I've kind of given you an overview of our native MS platform, for the next portion of this talk, I'll focus on 4 distinct applications where we found our online platform to be particularly advantageous for AAV analysis. The first of which is for the rapid screening of AAV samples. And here I'm showing that this method can be generically applied for the rapid screening of capsid compositions of different AAV serotypes. And if we do a quick qualitative comparison, we can see relative trends in the MS data align quite well with the respective empty, partial, and full capsid content ratios obtained by AUC. So for example, according to AUC, AAV1 should have higher partial capsid content compared to AAV5 and AAV8, which we can see here in the mass spec data. And also AAV5 should have greater empty capsid content compared to full capsid content. And we can clearly see that these trends are well reflected in our MS data. So next, our goal was to evaluate the sensitivity of our online method for capsid detection. So in order to do this, we diluted empty capsid material by increasing amounts and ran the same injection volume each time. So this low end of our detection limit reflects an estimated protein concentration, capsid protein concentration rather, of 1 microgram per milliliter. And this corresponds to a capsid titer of approximately 6e⁻10 capsids per milliliter. Moreover, we can see a very linear correlation between between injection amount and MS response within this evaluated range of 5 nanograms to 1,000 nanograms of total AAV capsid material injected on column. And overall, we believe that this suggests highly sensitive AAV capsid detection that's amenable to low titer formulations, therefore making this a compelling approach for monitoring in-process samples. So as a logical next step, we applied this method to samples derived from downstream processing. For this experiment, in process, AAV8 samples were analyzed from fractions obtained before and after an anion exchange polishing step. And we can see that prior to anion exchange, there's a significant level of empty capsid in the AAV material. However, after this polishing step, the abundance of full capsid is dominant in the spectrum, thereby confirming successful enrichment of our desired product. And I also just want to emphasize that the acquisition of this comparative data set was accomplished in about 20 minutes. And so this is really well suited for rapid screening of AAV material at critical stages of production, which can in turn be used to accelerate process development and optimization decisions. So next, we sought to evaluate how quantitative the MS response was for determining empty to full capsid content ratios. To do this, an AAV lot with known AUC capsid ratios were spiked into AAV empty control lot with varying volumetric mix ratios. And the fractional amount of full capsid in each mixture was quantified based on the relative area of the extracted ion chromatogram from the spectral region ranging between 35,000 and 50,000 m/Z, which corresponds to the signal of the full capsid species. And so, as you can see here on this plot, on the right, the native MS approach demonstrates excellent correlation with the industry standard AUC method for percent full capsid quantitation, which is indicated by this strong linear relationship between both methods with a slope approximating 1. Next, we wanted to evaluate quantitation for more complex AAV samples that contained varying levels of partially filled capsids in addition to empty and full capsids. So for both AAV1 and AAV5 samples shown here, we can see that the empty capsids are reasonably well resolved, whereas the signal from the partial and full capsids shows some degree of overlap. So as a more straightforward approach for obtaining semi-quantitative capsid content ratios, we use the inflection points in the spectrum to establish signal boundaries arising from the differentially filled capsids. And we use the extracted signal from these regions to perform the relative quantitation. And so when we compare our MS quant to the AUC-based quantitation for both AAV samples here, our MS approach shows some underestimation of partial capsid and slight overestimation of full capsid content. And we attribute this to the overlapping signals between these species. More advanced processing methods, so for example, peak fitting models, will likely improve the quantitation power of this method, but overall, this approach still aligns very well with the AUC-derived capsid content ratios. So finally, we explored how sensitive our platform is to changes in capsid stability. And so for these experiments, 2 different AAV materials were selected that show known differences in capsid stability under thermal stress. And they were subjected to incubation at 37 degrees Celsius for up to 45 days. And so in this first example here for AAV1 material, by comparing the relative abundance of empty versus full capsid across time points, there's an obvious decrease in the abundance of the full capsid over time, which suggests that it becomes increasingly destabilized over extended periods of thermal stress. Moreover, we can also see this shoulder peak that starts to emerge in the stressed samples. And while we can't readily discern what this is based on M/Z shift alone, it is unique. We can see that the shoulder peak is unique to only the stressed samples. By contrast, when we look at a highly stable AAV8 material, we observe very little change in the relative abundance of empty and full capsids over time, which suggests that the full capsid is largely impervious to thermal stress at 37 degrees C for at least 45 days. And so the differences in these full capsid stabilities between the AAV1 and AAV8 material may be related to differences in the 2 different serotypes, but can also be attributed to the relative size of the packaged genome, because there is some evidence that the smaller the genome, the more stable it is. But regardless, this nicely demonstrates the sensitivity of our online native MS method to changes in sample composition. And so just to briefly summarize this work, I'm highlighting several key advantages for our online native MS approach for the analysis of AAVs. And the first of which is that this method is really streamlined for high throughput native AAV analysis and requires no additional sample handling prior to analysis. This method also offers rapid quantitation of empty and full capsid ratios that are comparable to the analytical standard or the industry standard of analytical ultracentrifugation, but really requires a fraction of the time and the material that's required for AUC. We've also demonstrated that we're able to partially resolve information for partial capsids from the native mass spectrum, and we can provide semi-quantitative empty, partial, and full capsid content ratios. And we're currently working on ways to improve the quantitation of these species. And we've also demonstrated that this method is sensitive. It exhibits extremely sensitive detection, which is amenable to monitoring in-process samples. And finally, I just want to kind of highlight in this table here that our method compares favorably to other methods that are commonly employed for quantifying AAV capsid content ratios, but offers compelling advantages with respect to the limited sample requirements and rapid analysis times, as well as its commercial availability. And with that, I would just like to take a few moments to acknowledge those who've made this work possible. So first, many thanks to my colleagues in the analytical chemistry group at Regeneron, specifically our group leader, Ning Li, and my team leader, Shunhai Wang, for providing excellent guidance and support, and also just for creating an inspiring and science-driven work environment and culture. I'd also like to thank our colleagues in the viral production core for providing all of the AAV material that was used for these studies, as well as our colleagues in protein biochemistry who provided the AUC data that was used for all comparisons shown. Also, thanks to the AAV Analytics Working Group for always providing insightful and meaningful feedback and discussion. And lastly, I would like to thank the Newomics team for being such excellent collaborators and for providing the opportunity to present this work today. And of course, thanks to the audience for your attention. And I believe from here, we'll begin the final Q&A portion of our time today. So I would be happy to take any questions, and we can go ahead and get started there.
Okay, great. Thank you for that presentation, Victoria. It was excellent. I really appreciate it. All right, we do have a number of questions. So let me read the first one off to you here. Is the data presented using CDMS? I saw this come in the middle of your presentation. So I think maybe if you showed that schematic you had of the setup you were using for the LCMS, that might clarify that one.
Okay, let me go to this slide here. So you're talking, are you referring to
the one? Yes, that one. Yes.
This slide? Okay.
Yes, that's what the bulk of the data was acquired.
Yeah, so the bulk of the data was acquired using this method. So we haven't shown any CDMS data here. The CDMS data that I showed on, I believe it was the previous slide. So this is previously published CDMS data. None of the data shown here was acquired with CDMS.
Okay, all right, great. All right, question number 2. It appears that the ratio of your empty and full capsid changed as you added the charge reducing agent. Could you explain why this is? This
is a great point here. So for this is a very good catch. Okay, so for this sample here, we have our detection parameters optimized for the charge reduced samples. I just did a quick comparison here. So these are not optimized. This is suboptimal for the non-charge reduced parameters. So we can get a more accurate ratio if we optimize the parameters accordingly, but I just didn't spend the time to optimize the parameters for the non-charge reduced because this was really the focus of this work.
Okay, great. Another question. Do you see any evidence for aggregation of capsid particles? For example, mass to charge 60 to 80,000.
Yeah, so we do see sort of the tailing feature, but we believe, so some of these higher M over Z tailing features, but we believe that that's just sort of more extensive charge reduction. So maybe not fully controlled charge reduction, but we really can't, we're really not detecting anything arising from aggregation that we can readily distinguish from these data sets.
Okay, great. Do you have an assignment for the signals seen at about 45,000 mass to charge, the features above the assigned quotes full peak?
So I think that is what we were just discussing here. So what we think this is caused by is likely more extensive charge reduction because we can't fully control the charge reduction in the desolvation gas setup. So I believe that's where these tailing features are arising.
Okay, great, thank you. What is CDMS? What does that acronym stand for? CD-MS.
CDMS is charge detection mass spectrometry. So it's a it's a method that's really gaining traction now. It's a single ion technique. I don't want to go into the bulk of it because I'm not a CDMS expert, but I think that this is a really exciting area that holds a lot of value and a lot of power for looking particularly at extremely heterogeneous species. And so I imagine that we're going to see that technique really gain traction in the future.
Okay, great. Let's see. Do you see changes in the empty-full ratios as you change your MS source conditions?
Yeah, so we have tuned our source conditions here to the most quantitative outputs we could find. So we definitely optimized our source conditions to allow for the larger full capsid species to transmit most efficiently. So we've tried to optimize our front end conditions accordingly. And like I said, so early on when we started this work, we were not observing highly quantitative capsid content ratios. And we found that through tuning, more extensive tuning of the front end ion optics really enhanced our transmission efficiencies particularly for the full capsids. And so through extensive optimization, we have been able to achieve quantitative transmission of these different species.
Okay. What Orbitrap resolution did you use to collect these data?
So these data were collected at low resolution because in our case, we found that high resolution doesn't really do any good for us in this case because these are such heterogeneous samples. So in order to maximize our duty cycle here, we just used low resolution in this case.
Okay. Have you tried any other emitters with this technique?
So I initially started not the online. I did some AAV analysis using the static tip emitters and ran into a lot of issues. I think the issues may have been arising from the offline buffer exchange. And I think sometimes there are stability issues for these AAV particles when they're removed from their formulation buffers for an extended period of time. So I suspect that maybe there was aggregate formation or something like that over time. And so I often ran into spray stability issues using the static emitters and have really been able to overcome that using this online approach in conjunction with the M3 emitters.
Okay, great. So this one is to confirm you found that surface charge was very similar for full versus partial/empty capsid. Yes.
Yes. So in general, with this method, we can't fully confirm that, but we're basing this assumption based on the fact that we get good resolution between the empty and full capsid material. We can assume that the assumptions that are empirically derived from this earlier CDMS data hold true. And this is all based on CDMS data, the charge detection mass spectrometry data. But importantly, the front end, the native electrospray process should be consistent between our native MS approach and the CDMS approach because we're essentially using very similar ionization processes for our data sets versus these that were acquired via CDMS.
Okay, great. Thank you. You mentioned in-process support. Is there any sample prep/cleanup required for AAV culture before LC native MS analysis?
Yeah, this is a great question. So we've done some analysis further back where there was more culture components in the sample, and the signal to noise was not nearly as good, but really the front end buffer exchange allows us to look at these samples that are directly from in process without any sort of purification ahead of time. So like the polishing step, that's technically in part a purification step. And we were able to look at our samples prior to the polishing step and get really good signal to noise. So I can't tell you from the very beginning of process development to these more later stages, but we have shown that, later stages when we're sort of fine tuning the process, we're able to clearly, easily look at these in-process samples without any issue using our desalting column on the front end.
Okay, great, thanks. Two more questions. All right, so this one is apparently a reply to one of your earlier answers. Reply to your response of the 45,000 master charge assignments, : do these signals go away if you don't use the charge reducing agent?
Yeah, so they're not as defined, but even in this case here, this is a case where we're not applying our charge reducing setup, and you can see that there's a little bit of this tailing feature here. So just so happens that we're on a good example here. So I would say the under charge reducing conditions, depending on the signal to noise of the sample, this tail feature can become more defined. If it's lower signal to noise, we see that oftentimes this tailing feature is a little more defined. So I think it also sort of depends on the overall concentration that we're looking at and relative signal to noise. But you can see that even in this example where we aren't applying charge reduction, you still see a little bit of this tailing feature. So we're not convinced that it's entirely that this tailing is entirely an artifact that's caused by charge reduction. I just think it contributes to it a little bit more.
Okay, great, thank you. Two more questions. Does different modifier gas / solvent have the impact on empty versus full ratio measurement?
So I will say this: when you change the charge states, you need to tune your parameters a little bit more to get a quantitative response. So we have seen changes in capsid content ratios, but this is generally when we aren't optimizing our source conditions or ion optics, our transmission conditions for the charge states that we're looking at. So generally, when we've found that when we have we can generate a sort of generalized set of parameters that works well across all serotypes when we optimize for our charge reducing conditions. So it's not something that we necessarily have to tune to different serotypes or anything like that. It just seems to be impacted by overall surface charge. So I think this response can be tuned to be quantitative across the board. We just have to make sure that our ion optic conditions, our transfer parameters are optimized for the charge states that we're looking at.
Okay, great, thank you. Is online mass spec success to a certain extent depends on successful fraction of SEC? Question: how does the method detect partial if SEC can't separate partial from full?
So in this case, I guess something to point out. This is a great question. Sorry, I'm going in the opposite direction. Okay, so for this case, we're actually not using SEC as a separation mechanism for the AAV itself. So we found a desalting column that really just serves specifically as a desalting column. So all of our AAV species are coming out. There's no resolution for this particular desalting column for the different capsid components. So all of our capsid components are coming out at exactly the same time. The whole point of this front-end desalting column is so we can remove these non-mass spec compatible salts ahead of time. So we can, start to integrate things like a divert valve that can divert this salt peak away, and it's no longer going to go to our mass spec, to the front end of our mass spec, right? We can just divert this away. But we are not using SEC as a separation mechanism for the different capsid species. So the only separation that's occurring is in the M/Z space. It's not due to separation from the SEC column itself.
Okay, good. The questions are still rolling in here. So I know I said 2 a long time ago, but here we go. I think you might have answered this one, but can you also separate the particles with intact or defective capsid proteins in this technique?
Sorry, can you repeat that question?
Can you also separate the particles with intact or defective capsid proteins in this technique?
That's a great question. So I think that our thermal stress samples maybe point to our ability to detect changes in composition, but I'm not sure that, I think it kind of depends on the extent of the capsid composition. So if it's a minor change, we're not going to see things on the PTM level when we're looking at, shifts that might be caused by smaller PTMs or things like that. We just don't have the resolution for that at the intact level. But I mean, we are able to detect major changes that are occurring, at least in the relative capsid content. And so, you could see in the, I can pull up the example. Let me see here. So if we go back to this stability data set, one thing to just point out here is that, we start to see this shoulder species, and I'm assuming, this has something to do with either maybe loss of the genome information or something along those lines. We're not entirely sure. We can't resolve this information by M/Z alone, but you can clearly see that this is probably a decomposition product following thermal stress that we otherwise don't see in the non-stressed sample. So I hope that answers the question. I'm not sure.
Yes, thank you. I think it does. Can you use online IEC to separate the AAV species?
So I'm sure that we can to some extent. In this case, that was kind of beyond the scope of what we're doing here. This kind of goes back to what I was talking about before with the SEC separation mechanism. In this case, we're only using that to remove non-mass spec compatible buffer components prior to analysis. So we aren't necessarily, the objective, at least for this application, is not to separate these differentially loaded capsids. We want to see them all come off at the same time, and this separation is merely occurring in the M/Z space. So yes, I'm sure you can do plenty of front-end separations, and I think this platform is very amenable to that. That's just not the scope of the work that we did here, but yes, I anticipate that you can easily adapt this platform to do additional or alternative front-end separations, if that's your objective.
Okay. Great. Sophie, you have a question you wanted to ask? I see you're sending me a chat, but I'm not quite getting it. I think that's all the questions from the audience at this point. Thank you, Victoria, for a great presentation and handling many questions.
Thank you so much for this opportunity. This was a great experience.
Thank you very much for presenting and working with us.
Of course. Thank you. I just want to give you all a shout out. You know, our team has really been able to advance a lot of our online applications through the use of the Newomics Mensi platform. And we're very excited about new applications and what other applications are to come. So thank you very much, the Newomics team.
Thank you, Victoria. We really appreciate the collaborations with the Regeneron team. So it is a great opportunities. I hope other pharma teams can also collaborate and work with us to really advance precision medicine.
Yeah,
thank you.
absolutely.
Thank you, everybody.
All right. Thank you.
Thank you.
All right,
All
thank
right.
you, Victoria.
Bye. Bye. Have a great day. Bye.
Okay, bye.
Thank you. Bye.